means biosimilar developments are also, so many blockbuster biologics are also going off patent. So any biologics are going off patent, then we will be doing this biosimilar. Similar means we'll do the same drug. So same formulation we'll be doing. So that is called as a biosimilar. So after going to the after patent is going off, then we'll be doing that molecule, and obviously any pharma industry who is doing that molecule will be in a very good position. So that is the reason for biotechnology graduates are required there also. And other than that, and advanced therapies like mRNA vaccines and also therapy and then gene therapies, and there also biotech graduates are very much important. So these and all, but companies are also expecting other than requirement is there for the companies, but companies, as I said earlier, companies are also expecting you to bring some lab skills, means any previously some lab skills or any training courses what you have learned related to biotechnology, rather than they taking you as a fresher and giving training. So they expect at least some experience, at least internship experience, or at least 1 year experience in any biotechnology related field, so that for them it will be easier to take. So rather than joining as a fresher in the current market, I'm not saying it's a very difficult task or no. Fresher going into the industry, it's a little bit challenging compared to a person who is having any little internship experience or any industry related experience, at least for 6 to 1 year. So this is what we are seeing regularly in the current scenario. Um, that is industry readiness. That is what I was going to explain. Um, and one more topic, as I told earlier, I will discuss today is biotech a good career path. So biotechnology, it's a good career path, means pharma and biotech will always continue to grow. Because as I said to you earlier, in India is a global hub for vaccines. Whenever there is requirement, so there will be, whenever there is requirement, there will be opportunity. So whenever there is opportunity, that we should utilize it, and we should get into that. That means requirement is there, opportunities is there. So it's a good career path. So as I said, vaccines as biosimilars and new therapies are getting into the market. So for that requirements, they require biotech graduates. So biotech knowledge, because biotech persons know better knowledge uh, uh, in this segments like whatever discuss vaccines and all, because it's essential for this. They are essential to making these products safe and effectively biotech graduates. And coming to the growth realistic expectations, I don't want to so discussion means what why is there today we have kept a discussion is it want to show that the reality what is going on in the market. So whatever I have told you till now is the what is reality. It's not simply for a seminar purpose. I'm showing you any beautiful colors like that. No, what is going on in the market, I'm telling you. But coming to the salary and growth realistic expectations, your first salary may feel modest, but it's just a starting point. You cannot expect after your graduation, when you're going into any pharma industry, your salary will be very 5-digit salary and all. You cannot expect initially. That is what I can tell you with my experience. Because if you when you are comparing with our peers, means peers in the sense, when you're comparing with any software people, MBA, or any of your friends who are parallel to you, who are doing any other graduations, so we cannot compare with them, because the course we have chosen initially will be salary will be modest. I'm not telling modest in the sense, I'm talking about general point of view. So there will be few persons who will be getting a lot, means at least out of 10, maybe one will be getting, two will be getting. So that is not the average. Average is 8 people we are getting. What I'm getting is what I'm speaking. So that is the reality. So reality is opportunities are there, um, but you should not think after immediately after getting after graduation is finished. So you will be getting any your five-digit salary or more. More. I think you understood what I am saying. So your first salary, as I said earlier, your first salary may feel modest, but it's just a starting point. Going on in the career, can I can tell you frankly. So there may be software friends will be having, any persons who are working in softwares. Maybe they will be getting more salary. I agree, but what is the trend? There will be sometimes there will be layoffs. Software boom will be there. Sometimes it will go down. Sometimes it will go up. But till humans are there, till the diseases are there, pharma industry will not go down. So it will be in the constant level throughout our career. That is what I can tell from industry point of view. Um, and some some specialized roles will be there, like specialized roles for biotech graduates, like very few will be having. So like biostatistician or process engineers or medical science lesions or regulatory affairs, who are particularly very few posts will be there. Maybe they will be having more demand. So but whatever your salaries you will be getting, it will rise with the experience. So regarding that, I can tell you. And so other than this, what I want to tell is, this is what I want to tell you everyone. So other than that, if you have any particular industry related questions, or how it works, or any particular in department related questions, or which field to choose, or whether how to go forward, any other related questions, topics you will be having, I am happy to answer. And after this is finished, finally I will take some question answers, that is in the last. But before that, any particular related topics you want me to explain, I can explain you. So yes, please.
Sir, if possible, could you describe your job role in a formulation department? It would be helpful for us.
Sure, sure. So formulation R&D, um, it works from, as I want to tell, R&D is the heart of any pharma industry. So any development will start from R&D only. So coming to the tablets, we'll be having a different cycle, and coming to the injections, it will be having a different cycle. So any R&D tablets and all, what we are doing, it's an ANDA, abbreviated new drug applications. So RLD will be there. RLD is a reference listed drug. So that RLD is reference listed drug, means that is innovator who has developed that drug. So that is called as RLD, reference listed drug. So accordingly, after that patent is expired, we will be doing the same tablet, that we called as ANDA. So abbreviated new drug application. So it starts from whatever API, active pharmaceutical ingredient we have chosen, and whatever raw materials we have chosen, that compatibility studies we will do, and different formulations we will do, and we'll match our formulation with the reference listed drug. So matching the formulation with the reference listed drug is not a easy process, because any tablet if you are taking, if the RLD means reference listed drug, reference product, if it is taking 15 minutes to dissolve the tablet, so our product also should almost take the similar time. And if it takes 5 minutes to disintegrate, and our tablet also should almost take the 5 minutes to disintegrate. So these all we will be matching with the reference listed drug. Technically, if I go deep into that, you won't be knowing the technical terms. So I will be explaining in an easy way. Um, so whatever a tablet, any some few tests will we will match the tablet to tablet, means RLD reference product, how it is behaving. So we will also do our formulation in our R&D, which is almost similar to the reference listed drug. So whatever reference product I'm speaking. So we will, after doing the formulation, we'll give it to analytical department or QC. If it is in plant, there they will be doing the chemical analysis for that. So assay, you will be knowing assay, what is assay, how much water content is there, and dissolution they will be checking, and impurities they will be checking, related substances, and micro related they will be checking, and again in the TAMC, TYMC they will be checking. So weight of the tablet they will be checking, description they will be checking. Means quality, these are all quality control tests. Formulation R&D, as a working in the formulation R&D, will prepare a tablet. So that whether the tablet is having the quality parameters and all, they will be checked by quality control department, which we call analytical R&D in R&D. So planned it is quality control. So after that we have developed the product. So that product, what we will do. So it has to be, as I said earlier, we have made the product which is almost similar to the reference product. So whether both are behaving same in when it goes to the, when any patient is taking, so that we need to check now. So for that we will be doing a bioequivalence studies. So our product, what we have developed, and the reference product, what we have taken, so both will be given to the patient. There will be doing clinical trials. So these two tablets given to the patient separately, and at predefined intervals, we will be taking samples from the body, and we'll be checking how much drug is dissolved. Suppose 10-minute sample they will take, 1 hour they will take. Suppose 12 hours they will take, 24 hours they will take. So whatever our drug is releasing, and whatever reference part is releasing should be almost same. Almost same means some criteria will be there for that. So for bioequivalence and all criteria, so that percentage limits will be there, some 80 to 125 percentage and all. So if it is in that, with that limit, our product, we can say it is bioequivalent with the reference product. So if the after product is bioequivalent to the reference product, then we will file the product to the agency. And again, agency, it varies from country to country. US is having US FDA, and UK will be having EMA. Um, like Australia will be having a TGA. Different countries are having different regulatory authorities. How India is having CDSCO. Like different countries will be having different authorities. So our product is developed, we need to check stability also. We will keep in stability. So stability will be having different conditions. So accelerated condition will be 40 degrees centigrade, 75% RH, and long term condition will be 25 degrees centigrade, 60% RH. And this stability and all conditions will come from ICH stability guidelines. If you see the ICH guidelines, and that stability guidelines will be there, you can see. And stability, it's not accelerated means it not about only 40 75, and long term is not about only 25 60. So it depends on the product, and depends on the storage of the product, and depends on the container what we are choosing. Stability conditions will vary. So this is the means basic I'm explaining. So if you see the guideline, you will be knowing better for which condition, or which storage condition, which stability condition we should be going, and which type of pack, which stability condition will be going. So after stability is good and product is bioequivalent, then and all our quality parameters are meeting, then we'll give the product to the regulatory affairs team with all this data. So with all this data, regulatory affairs team will file the product to the concerned agency. So agency will be coming back. If this will review the our filing, ANDA. Will review the document, and if it is having any queries, they will come back. And if it is not having any queries, they'll give the product approval. So if product is approved, then it goes for commercialization. So as you know, commercialization will be done in a large scale, in a plant level. So from R&D now, from R&D, R&D's activity is finished, and now it goes to the plant level. So plant level, we will be having production team. Production will be manufacturing the tablets, and QC team will be analyzing the tablets, and QA will be maintaining the quality. And here, then batch will be commercialized, and it will be going to the patient, to which country we have filed. So in that country, we will be marketing our tablet. So this is how the cycle of that tablet it goes. Similarly, injection is also a similar type of dosage form, and is that it's a sterile dosage form. So much controls, and so much precautions should be taken, because it goes into the blood. So it is a very critical product whenever we are dealing with any injections. So injections also in a similar way, we'll be doing any for formulation, and then based on the bio requirement, as injection simple injections or solutions. Means our reference product is also a solution, ours is also a solution. In that case, there is no need to do bio. So depends on what type of formulation we're going. In case of simple solutions, there is no need to do bio. So if there is no only exhibit based on the batches, we need to show three exhibit batches to the agency. So with those three batches, we will submit our product to the agency, and if it is approved, directly we can do the commercial. Coming to the injectable suspensions or emulsions, so there will be giving the two options whether we need to do bio or in vivo characterization. So based on our feasibility and our requirement, we will be doing bio or in vivo characterization. So this is how injectable goes. And almost any pharma industry which are dealing with vaccines or tablets or capsules or injections will be going through the same cycles, because it's not a company specific or a person specific. It's a agency country requirement. It is so people come like that. We will develop the product, we will file the product, and we will commercialize the product. So that is that's the life cycle of any pharma product, like how it goes. think I think you understood at least a basic knowledge on that, because if I speak more technically and technical terms, you won't be understanding. That is the reason I have explained you in simple terms, so that you will be understanding how in the industry works, and how product from initial stages of development to filing and commercialization takes place, and what all departments are involved in it, and what is the role they are doing. I hope everyone is clear with it. Or if you're having any doubts on that, you particularly you can ask.
Good evening, sir.
Good evening.
Sir, I'm Pannaga. Sir, I had one doubt related to pharma companies and the salaries. As dry lab mostly deals with computers, is the salary for the dry lab roles usually more than the wet lab roles?
No, no. What you have asked, I understood. It's not like that. How nowadays, I have one more before answering question. I want to make it clear. So now, what I'm observing from at least from past 5 6 years is very people means that is what my observation. I'm telling, maybe your perspective will be different, because what I'm seeing is what I'm telling you right now. From past 5 6 years, most of the people, as you said, most of the people are interested in dry lab, means computer work or regulatory affairs or filing related activities. So they want a soft color job, and they want to sit in front of system. I'm not pointing anyone, I'm not discouraging anyone here. They want to sit in front of system, and they want to work rather than coming into the wet lab, because as I feel, pharma R&D, research and development person should go into the lab and should do the activity which is related to the lab. Means it is any wet lab or making a tablet or making an injection or making a solution or any titration or any liquid mixing. That is what core R&D means to me. But nowadays, most of the people are interested to sit in front of the system and do the soft color job or a system job, like how other software employees are doing. But comparatively, there will be no salary difference between who is working in the laboratory in front of any chemicals, and who will be sitting in front of a system. That entirely depends on our perspective and where our interest lies.
Okay, sir, understood.
Yes, please.
Sir, I have noted that the formulation department shares the MOA to the analytical department to perform certain tests. How does the formulation team come up with the parameters that needs to be checked, like assays, dissolution of stability? Is there a standard way to decide this?
Yes. So it's a good question. Going to the from formulation to analytical. So coming to the tablet, capsule or any oral solid doses from point of view. So we'll be doing, so, and if you suppose one tablet is there, any API name you can take into consideration. So I'm asking you people only to decide any API. what daily any you are seeing any drug, you tell me one drug name, I will take that example and I'll tell you. Any tablet name you will be daily using in the market or you have seen it, you tell any name, with that example I will explain you, it will be more easier. Okay, paracetamol, Panaga Ravindra has shared. Very good. Suppose paracetamol is Dolo 650. So Dolo 650 is the tablet paracetamol we are referring here. We'll think that is a reference product. So the paracetamol Dolo 650 was prepared by some X company. That is the innovator. What I'm telling in the now, I'm referring paracetamol Dolo 650 as an innovator. Means he is the first to first to discover or means to manufacture this API. So after the patent was expired, now Y company I came and I want to prepare the paracetamol with a different name. So now what I will do, I also have prepared a paracetamol. So whether paracetamol I prepared, actually it contains paracetamol or not, how you know? That is the identification test. One is identification. We'll be telling QC or analytical team you just identify whether paracetamol is there or not. So paracetamol will be having some particular having some characteristic peaks or characteristic test when we react with some any material. So we'll tell them to identify whether it is paracetamol or not, what I have prepared. Secondly, I am paracetamol, you're telling paracetamol 650 mg. So whether 650 is there in the tank or not, who knows? So I'll tell them, you check, I have prepared, as a formulation scientist, I prepare paracetamol 650 mg. You check the assay. So I'll tell analytical team to check the assay whether paracetamol 650 mg is there or not. If I didn't add 650 mg by mistakenly I add 400 mg only, then analytical persons will tell, will they report it contains only 400 mg. So when it's going to the market, it should contain 650. That is the quality. So I will tell them to check the assay. And other than that, I will tell them to check the water content in that. Why water content I'm giving tablet is a solid dosage form. So any tablet, any material or any formulation, if water is present in it, there is a chance of growth of microorganisms. Correct. So there should be limit for water content. So whatever water content limit I kept, whether the tablet is within that limit or not, third I will check. So I told them, I check the identify whether paracetamol is there or not, and secondly whether it contains 650 mg or not. Third, whether it contains water within the limit what I have told. Okay, then paracetamol. I have one more, three are finished. Next test, what I will give them is the dissolution. Okay, I have given paracetamol. I have taken reference of Dolo 650. Dolo 650 is dissolving 80% of the drug in 15 minutes. Sorry, suppose 85% of the drug in 15 minutes. So my tablet, I have given to check whether my drug is also dissolving 85% in 15 minutes or not. Because if my drug is not dissolving 85% in 15 minutes and it is taking 45 minutes to dissolve, then whatever formulation, whatever tablet I have done, it's not similar to the Dolo 650. That is wrong. So I will tell to check the dissolution, whether my drug is also dissolving 80% in 50 minutes. So the dissolution is finished. Then based on the criticality, sometimes I'll give the disintegration test. So the disintegration, if the tablet of Dolo 650 is disintegrating 15 minutes, might not more than 15 minutes, mine also should disintegrate within 15 minutes. So I'll tell, okay, you check my tablet also, check whether the disintegration is happening within 15 minutes or not. So 5 tests are given. Other than that, these and all are there. Impurities will be there. Any API, any active pharmaceutical ingredient, during manufacture of API, impurities will come. And during formulation also, process impurities will come. And during manufacturing also, impurities will come. So whatever impurities are there, so that impurities are having some guidelines. So impurity, ICH impurities will be there. ICH impurities. So ICH will be telling impurities and their limits. So accordingly, my drug will keep some specifications and you check for impurities in my drug also. So they are checking for impurities. Other than that, micro tests also they will do, because TAMC, TYMC they will be doing in the microbial growth or not. They will check within the specification limit are there or not. They will check. So these are the eight examples I have given. So these tests and all, and weight of the tablet they will check. So whether I'm compression, I'm doing tablet compression. So 650 mg paracetamol I'm adding. Other than that, only with paracetamol we cannot compress a tablet, right? So we'll be adding binder, we'll be adding super disintegrant, we'll be adding lubricant, So these all contain, if you mix it, tablet weight becomes around 800 mg, 850 mg, 900 mg. So I am telling the QC analyst 80% to check whether weight of the tablet what I have given the specification is meeting that or not. So these and all, again, it is not my requirement. It comes from the USP guidelines. So United States Pharmacopoeia general chapters will be having particularly for tablets what test we will be doing. So the tests and all we are covered or not, based on our product we will be doing these tests and we will be giving them. So each specific test will be having each specific specification depending on the country and the market we are filing. So accordingly, these test this limits, test specification, test limits, and what test to do, will be giving to them, and they will be doing, and they will be giving the output. So whatever formulation or whatever tablet I have done similar to Dolafusix 650, some formulation I have done. So the tablet is same or not, we'll check. Then, as I said earlier, we'll go again, we'll check for bioequivalent studies in the humans, and then we will file it. So like that, tests are decided. Same for parenterals also. There is a USP guideline, USP one I remember, USP general chapter one for parentals. Whatever tests are there in that, so those tests we need to be doing. For example, for injectable, they will their resolution won't come, because if I'm talking about simple injections, simple injection resolution won't come, and it's not a tablet. A disintegration also won't come. There, what it comes is description will come, pH will come, SA will come, and RA substance will come. Other than that, it's a sterile dosage form, microbial test, and sterility test will be there. Sterility of the dose should be sterile. Whatever formulation we're doing should be sterile. Bioburden and all will be doing. So all related to sterile, and whatever basic test will be there, that and all will be doing, and we'll be telling to the agency. I think you understood by the example what test we'll be giving and why we are giving.
Yes, sir, I got it.
Yes, please.
Hi, sir. I'm Roshna. Sir, so looking at like the market perspective, China is releasing a lot of innovative products, and seeing that even India has a lot of workforce and a lot of research going on, where do you think that we're lacking? Is it the research part, or is it the funding part, where or the regulatory part? Where are we going behind?
Uh, um, China, what you said, China has so many innovative, I believe it is not releasing so many innovative products. China is releasing so many, means whatever is there in the market, it is doing A and A. What as I said earlier, like suppose dollar 650 is a reference, we are doing one tablet similar to the dollar 650, correct? So like that, similarities or whatever we call it, the terminology as A and D, abbreviated new drug applications. After the patent is expired, those all formulations and all they are doing, and APIs, but particularly speaking, active pharmaceutical ingredients has increased 10 times, tenfold. API manufacturing has increased from the last 5 to 6 years, and in China. What you said is correct, but not about innovation, and it is about more APIs are coming from China. Uh, why they are doing it? Because I have seen so many pharma companies these days, they are taking a lot of APIs from China. Why? Because the manufacturing cost and that cost will be very less. Any Chinese product will be getting, as you know, in India or any other countries, most other products are made in China only, because their manufacturing cost is very less. So in that way, China is manufacturing a lot of APIs. You won't believe how many APIs they're manufacturing. Suppose that API is in manufacturing site is in India also, we are buying it from China, because they are giving it for a very less price. So that is a very uh, as you know, this is a business. Pharma company, end of the day, will be employees, but for a pharma owner, it is a business for them. So if the any material, if a raw material is getting for a lesser price, obviously we'll choose that API which is coming from China. And it doesn't mean that quality is not compromised. So quality will be there. Otherwise, they won't be taking that material, because we will be filing it to the any regulatory agencies for US and UK, which are very strict in their regulations. So that APIs, whatever we are getting, it's very cheap. That is the reason we are taking it from China. Hello.
Yes, sir. Thank you.
Yeah, did I answer your question or anything is there?
Yes, sir.
Thank you. Thank you. Any other questions are there? Yeah, yes, please.
Hello, sir. Sir, I have one more question. Sir, as you yourself have completed a PhD, I wanted to know your perspective regarding this matter. I attended one guest talk, and a person who was giving the guest lecture, he kind of mentioned that when someone completes a PhD, the area of research becomes narrower because a person will be focusing on a particular part of the larger subject. And
Correct.
when it comes to the job prospects, uh, it will be like he or she will be having a disadvantage. Do you agree with this, or like how do you see this? Completing PhD and getting into job, or completing masters and directly getting into the job?
Yeah, understood question. So my experience on this is, as I did the PhD, so I am also not discouraging anyone to do the PhD, but my suggestion to do PhD for everyone, whoever wants to do PhD, so they should do part time. That is what I recommend, because we will be, yeah, I won't be telling we'll be wasting, but anyone who is having experience in industry for 5 years and who came as a fresher, with PhD won't be getting a package, won't be the same who is having already 5 years in the same field. So that is what I believe, because I have seen on all 10 plus years in the industry, directly coming into the industry and doing part-time PhD is better rather than doing full-time PhD. This is what my suggestion is. And narrowing particular research work is, it's not, I may not feel that is correct, because wherever you are working, that depends on the work what you are doing. As I discussed, what we'll be doing daily routine in our pharma labs and all. So that doesn't narrow a particular segment while we have done the PhD. So if we are having the PhD, our research, our experience, our PhD experience will help us to do the research activity more when compared to the normal persons. That is what I feel. But PhD won't degrade, or it won't narrow any search criteria for you. And my suggestion on PhD, as you said, is better to do a part-time PhD rather than doing it in the full time, because your friends, over your friends will be joining a job after your post graduation, and some you will be going to a PhD. So after 5 years, he is already having 5 years of experience in the same industry. So after you 5 years, if you go into the industry, you are still a fresher, because you don't know how industry works. You are having a PhD. I agree with it, but how industry works, you won't be knowing compared to what your friend or colleague will be knowing. So that is what I feel regarding the PhD. Excuse me, and the industry.
Okay, sir. Understood, sir. Sir, if I can ask one more question.
Yeah, yeah, please.
Sir, as I think, sir, I was looking at your LinkedIn profile the other day, and I saw that you worked as a manager in a company. Sir, I wanted to know, as we are students, when we approach companies for internships, how does people who are already working in the company want them to be approached by the students?
It's a good question. Generally, internships in the company will be very limited. Suppose in formulation R&D, maybe one teams, two team will be there. So each team will be having maybe one vacancy or two vacancies. So other team will be having one or two vacancies. So total of four vacancies they will be having. Means most of the time, more than that, means like how employees will be there 20 30 employees, same in the same manner. They cannot take 20 30 interns. So whoever is approaching any person means will be some persons will be approaching HR, and some persons will be approaching through any contacts. Some persons will be approaching through any of your colleagues who are working in the same industry. So it depends. It depends on your profile, on your interest. Some how we look into the persons who are coming for the internship is whether they are seriously working the pharma industry, or some colleges and some universities require internship for their course completion. So by the interview, we will get to know whether they are serious about what they will be doing, because because I know who are into internship, and if they have done very good, and we have absorbed them. So after a completion of internship for 8 months or 9 months, we have absorbed them to as an employee, because they have the interest to work, and that is what we see. And there are some people who will be coming particularly for a certificate requirement, or their college requirement for them to do an internship. So those kind of people will come, they do their activity, and they will be going. So accordingly, based on the requirement, we'll be going forward with the interns.
Okay, sir. Sir, should we approach people through LinkedIn? Like, should we text them if there are any open positions, or should we mail them? What is the better approach?
LinkedIn is a what do we call the exact terminology? It's a job related, and it's a good platform, and it's not like any Facebook or Instagram. So whoever you want to send a message, or whoever you want to discuss with, you can send a message in the LinkedIn. That is not at all a problem. So LinkedIn, primarily it is there for everyone to connect regarding their jobs, and their professional manner. So if you are messaging anyone in the LinkedIn for any job opportunity, that is not at all wrong. So you can approach them, and you can proceed, because it's a professional platform for job related activities only. Other than job, means any professional related activities, filling LinkedIn is there. So other than that, if you are having any contacts in the industry, you can approach them, because what I can tell, see, I don't want to frankly show you what is not reality in the market, because people, so many, any job vacancy, if any bigger company is posting, there are a lot of applications, and there is lot of demand for any post. So due to the demand, most of the people won't be getting in the area they have chosen. As I've discussed earlier, and during my first initial 5 minutes, nowadays most of the people I'm not seeing who has done graduation or post graduation in biotech or any pharma. So they are going out of their fields due to job opportunities, and maybe lesser opportunities, or whatever particular department they want to choose, they may not be getting. That is what I feel.
Okay, sir.
Yes, please.
Sir, I've been considering shifting towards pharmaceutical science, especially for my PhD. So I wanted to ask from your experience, how possible is it to make that switch? Or if it is possible, can I do it in India? Or as you said, since the APIs are being mostly produced in China, or is it good to do in China or any other countries?
Switching from biotech to pharma, it's not at all a difficult task, because more biotech and pharma the related subjects and what we'll be studying is almost similar. So that is shifting from biotech to pharma is not a difficult task. But my question to you is not about PhD. So after completion of PhD, how you want to go forward is my question first.
I would like to work in the formulation department.
So that is what I have said to our friend Pannanga Ravindra previously. So if you want to work in a formulation, that there is no requirement for a PhD primarily. So you can work in any formulation R&D formulation department, and simultaneously you can do any part-time PhD is what my suggestion. And you, as you said, you want to go to any other country and do a PhD. So as you also know, and everyone will be knowing, uh, not about China. China study wise and all, most of the people won't prefer there, because it's very difficult to study in China. I think it will be in Chinese. They won't be translating in English. That is what I know. Because what Chinese, they will be speaking only in Chinese. They won't change it. And not about China, any other country, if you see, you'll be knowing now in US US is also very difficult to go, because jobs and all are not there. There so many people are coming back. So if formulation job is our priority right now, I suggest you to go into the formulation R&D job, and then, and as I suggest you to do the PhD part time in any reputed university in India. So that is what I can suggest you.
Okay, sir.
Because I was from initial onwards, I was telling the same. Rather than doing a full time, my suggestion is, rather than doing a full time PhD, you do a part-time PhD while doing any job. It will be better for your career growth, and simultaneously you'll have a doctorate also with you. That is what my personal suggestion is for everyone, whoever wants to whoever is confused regarding that.
Yes, sir. Thank you.
And any other questions are there? Can ask any related question related to industry or jobs. Means means and first of all, how many of you want to be in the core department? Means what you have studied biotechnology and all, how many want to be in the core department? See, very few hands or a few hand raised, because so other people want to go to MBA. Good. Because I know you also will be knowing. You also will be knowing, not rather than me explaining, you'll also be knowing how many of your seniors who did this biotechnology graduates, and they are in the same field. Means they will be going, any of your friends and seniors, and they went into MBA or bank jobs, or any other job related to other than this our field. See, it will be there. That's what I'm telling. So many will be there, because frankly speaking, we are into science group, and I love science group from my childhood onwards. Means everyone who is into science group will want, as a primarily we want to get into MBBS. We didn't get the MBBS, so we came in this field. So science is my passion. So I went into the R&D, and now I am doing research. So any other software job now, if you give me any 6 digit, 7 digit, and tell me to sit in front of system and do software job, means I cannot do that, because I want to be in the laboratory. I want to do research, and I want to be like what I am doing now. That is how I feel happy. That is, other than, if you are not happy, we cannot we cannot work, because daily it's not a one day activity. College, your college activity maybe 4 years, 5 years it will be finished. If you are bored of college also, you cannot at least in 5 years it will be 100% finished. but after that you should do the job. It's not a one day 5 years activity. At least 20, 25, 20, 30 years you need to work in the same industry. So without interest and without dedication, we cannot work. That is what I feel, and I want to share that with you.
Uh, Sir, I have one more question. Okay, Hitanshi, go ahead. Hitanshi, go ahead. Yeah.
HPLC. Simple, simple. If you want to go into analytical, you should have a hands-on experience on HPLC. HPLC is the primary equipment which will be used in the analytical. Other than that, GC will be there, LCMS will be there, HRMS will be there. These are all are sophisticated experiment instruments which maybe during your course of working in QC you will be knowing. But HPLC is the primary equipment, primary instrument you will be you should know before getting into the QC. And one more thing, as we said, QC. I want to tell you, QC quality control will be in the pharma industries. So any pharma industry is QC job is a standing job. You know that. As I wanted to say whatever is in there in the pharma industry, I want to frankly to explain to you that is my motto today. So I don't want to show what is not there. I want to show what is going on. Should stand for at least from 5 to 6 hours in that QC means. You should be prepared for that, and you should learn HPLC. Then you'll get into the QC. Your interest is in QC or analytical R&D. Analytical R&D will be in R&D. Okay. Yeah, QC means it will be QC will be in the quality control will be in the plant level. So plant level means GMP will be there and so much regulations rules and all will be there. But analytical R&D is in the R&D level. So what we call formulation R&D is in R&D level, and in plant level it is called as production. So bigger scale we will in R&D